So, good morning all of you. Welcome to the online botany classes. In this video lecture, I will discuss about cell cycle. So, let us start. You know what is cell division? Cell division is the process in which new individuals are produced. Okay. Cell division not only produces new individuals, it also helps in replacing the old and damaged cells. Got it? So, this is what your cell division. You have quite an idea about what cell division is as far as definition is concerned. Now, what is the cell cycle? Cell cycle actually consists of sequence of events, okay, which occur between two successive divisions. So, cell cycle involves sequence of events that occur between two successive divisions. Between two successive divisions, all those events that occur, they constitute cell cycle. Okay. Cell cycle involves two very important phases. What are those? Interphase, otherwise known as resting phase, and M phase. Okay. Interphase, otherwise known as resting phase, and M phase. Now, the cell growth and the DNA replication occurs during this interphase stage, and the distribution of daughter chromosomes occur during the M phase. Okay. So, as far as interphase is concerned, the cell growth and DNA replication occurs. We will discuss it in details in the next slide. And in the M phase, it involves the distribution of daughter chromosomes. Okay. Now, as you can see in this picture, this is a diagram of cell cycle. The cell cycle involves interphase, this G1, S and G2 phase. These three phases constitute interphase. Okay, these three phases constitute interphase. Okay, we will discuss one by one what is G1, what is S and what is G2. Then after this interphase, there is presence of M phase, where distribution of daughter chromosome takes place. Okay. Now, let us come to the G1 phase, or first interphase. Then within that interphase, we will discuss about G1, S and G2. What is interphase? Interphase is the period between two successive divisions. Okay. It is metabolically the most active stage, but it is otherwise known as resting phase. Why we call it resting phase, that I will tell you. So, it is the period between two successive divisions and metabolically the most active stage. It is metabolically the most active stage. And remember one thing, it is the longest phase in cell cycle. In cell cycle, it is the longest phase. Okay. Here, what happens? Here the chromosomes decondense and are distributed throughout the nucleus. Okay. In the process, during the interphase stage, the chromosomes decondense. Whatever the condensed chromosomes were there, they decondense. Got it? And are distributed throughout the nucleus. Okay. The interphase is completed in three steps: G1 phase, S phase and G2 phase. Okay. And remember another thing, interphase is otherwise known as resting phase. Why we call it resting phase? Because actually division of cell does not occur in interphase. However, it is metabolically the most active stage. As division is not occurring, we call it as resting phase. Got it? Now come to the G1 phase. G1, that means first growth phase, G1 phase. It is the longest period in cell cycle as compared to G2, S and others. It is characterized by increase in the size of the cell. Okay. In this phase, the cell size increases. And what are the events that occur during the G1 phase? It involves protein synthesis, RNA synthesis and various enzyme synthesis which are required for DNA synthesis. Okay. Protein synthesis, RNA synthesis and enzymes that are required for DNA synthesis. So, the enzymes which are required for DNA synthesis are also synthesized during this G1 phase. Got it? Well, as far as some animal cells are concerned, like nerve cells or your skin fibroblast, etc., they cease to divide. Okay, they cease to divide. So after G1 phase, they immediately, instead of going to S phase, they immediately enter into G0 phase. Okay. Here they remain metabolically active, but they are no longer dividing. They do not divide. Metabolically active, they remain metabolically active in the G0 phase, but they do not divide unless they are stimulated by external signals or extracellular signals. Got it? So as I have told you, G1 is the longest period in the cell cycle. It is characterized by the increase in the cell volume or cell size. It involves protein synthesis, RNA synthesis, and the synthesis of enzymes required for DNA synthesis. Got it? And in some animal cells like nerve cells, your skin fibroblasts, etc., the cell, after exiting from G1, or when the cells exit the G1, instead of going to the S phase, they go to the G0 phase. In this G0 phase, they remain metabolically active, but they no longer divide unless they are stimulated by extracellular signals. Got it? Now come to the S phase. S phase is otherwise known as synthetic phase. What occurs? What kind of substance is synthesized during S phase? DNA synthesis occurs. Okay. So during the S phase, DNA synthesis occurs. Thus, the amount of DNA per cell gets doubled, or DNA replication occurs. Okay. DNA synthesis or DNA replication occurs, as a result of which amount of DNA per cell doubles. Chromosome number remains same, but the DNA amount gets doubled. Okay. Each chromosome has 2 sister chromatids, and thus with double the amount of DNA present in original diploid cell. So in this phase, each chromosome consists of 2 sister chromatids. Got it? So after S phase, the cell enters into G2 phase or second growth period. Okay. What happens during G2 phase? Synthesis of proteins for spindle fibers occurs. So in this phase, the proteins that are required for the formation of spindle fibers, which we will study in details during the M phase, those proteins are synthesized in G2 phase. Okay. In case of animal cells where centrioles are found, these centrioles duplicate. Okay. In case of plant cells, no centrioles. So in case of animal cells, the centrioles duplicate. Got it? So this is what happens in case of G2 phase. So this is the concept of cell cycle as far as interphase is concerned. So interphase is the resting phase. However, it is metabolically the most active stage. It involves G1, S and G2. In the G1 phase, what happens? The cell volume increases. Various processes such as RNA synthesis, protein synthesis, and the enzymes that are required for DNA synthesis are synthesized during the G1 phase. In some of the cells, animal cells like nerve cells, the cell after completing G1 phase enters into G0 phase, where cell remains metabolically active, but it does not divide unless some external signals or external stimulation is given to it, extracellular stimulation is given to it. After G1 phase, cell enters into the S phase where DNA synthesis occurs. Then it enters into the G2 phase where proteins required for spindle fiber formation are synthesized. Okay. So this is what your interphase. So after the cell completes the interphase stage, it enters into the M phase. That we will study in a separate video about M phase. So let us know what are the checkpoints of cell cycle. You know checkpoints. What are checkpoints? Well, as far as checkpoints are concerned, your cell cycle has many checkpoints at different phases. What it does, what the checkpoints does, it prevents the entry of cell into the next phase if the previous phase has not been completed. Okay, has not been completed. If the previous stage has not been completed in a proper way, it does not allow the cell to enter into the next stage. Okay. Like if DNA damage has occurred, there is DNA damage checkpoints where at G1, S and G2 phases. Got it? At G1 checkpoint, what happens? It ensures repair of any DNA damage before its entry into S phase. So, the cell checks through the checkpoint at G1, whether DNA damage has occurred. If at all DNA damage has occurred, it repairs it. Then only it enters into the S phase. Got it? Similarly, there is S phase checkpoint. What it does? It ensures continuous monitoring of DNA. Okay, it ensures continuous monitoring of DNA to promote repair of any errors that occur during DNA replication. Okay, S phase checkpoint ensures continuous monitoring of DNA to promote repair of any errors that occur during DNA replication. Okay. Then comes G2 phase. Now, G2 checkpoint. As far as G2 checkpoints are concerned, it prevents initiation of M phase. Okay, the G2 checkpoints prevent initiation of M phase until the genome is completely replicated. So, G2 checkpoint prevents initiation of M phase until the genome is completely replicated. Got it? So, these are the various checkpoints in case of cell cycle. Okay. Now, let us discuss about the proteins controlling the cell cycle. Whereas, as far as cell checkpoint controls are concerned, at least 3 classes of proteins are found which control the cell cycle. What are those? Sensors, transmitters and effectors. What sensors? As the name suggests, sensors means it detects something. What? Something. That means the sensor or sensor proteins, they detect abnormalities and emit appropriate signal after that. Once it detects abnormalities, it emits appropriate signals. Now, the transmitters send the signal along proper pathways within the cell. Okay, and the effectors respond to the signal and inhibit the cell cycle machinery. Okay, so the 3 classes of proteins that control the checkpoints are sensor proteins, which detect the abnormalities if at all and emit appropriate signals. Then second one is the transmitters, which send the signal along proper pathways within the cell. Third one is the effector, which respond to the signal and inhibit cell cycle machinery. Got it? So, let us study about the functions of cell cycle proteins. So, what the cell cycle proteins do? If in case there is failure of surveillance system, what is this surveillance system? This particular checkpoints. If the checkpoints fail, then cell obviously progresses to the next stage of the cell cycle, isn't it? Suppose the checkpoints in case of your cell cycle fails, that is surveillance system fails. So, cell progresses to the next stage of the cell cycle. Okay. So, when a sensor protein detects the presence of DNA damage, you have heard about the sensor protein. So, what sensor protein does? Sensor proteins detects the abnormalities and emit appropriate signals. So, here the sensor protein detects the presence of DNA damage and triggers a response that temporarily arrests the further cell cycle progress. Okay. Then this delay is used by the cell to repair the damage. So, if at all the surveillance system fails, the cell obviously progresses to the next stage of the cell cycle. In that case, the sensor protein detects the abnormalities in DNA or any damage in the DNA and temporarily arrests further cell cycle progress, and this delay is used by the cell to repair the damage. So, a bit of delays occurs, and during this time or within this period, your cell repairs itself, repairs the DNA. Got it? It is very important as cells that undergo division with genetic damage may get transformed into cancer cells. So, if there is no such sensor proteins of these kind of things do not occur, then there is every possibility that damaged genetic materials get transferred to the next generation. So, it might cause cancerous cell. Got it? If DNA is damaged beyond repair, if it cannot be repaired, then the checkpoints in higher eukaryotes transmit a signal that leads to cell death, removing any possibility that the cell might become cancerous. So, there is provision that surveillance system is there. Suppose the surveillance system fails, then your cell cycle check proteins they play a role. Suppose the DNA damage has occurred to such an extent that they cannot be repaired. So what will happen? The checkpoints in case of higher eukaryotes transmit a signal that leads to cell death, as a result of which any possibility of formation of cancer cell will be removed. Got it? So there is any possibility will be removed. By how? By your transmitting signal that leads to cell death. So this is what the function of cell cycle proteins are. So let us let us have an idea about the cell cycle and checkpoints. As you have seen here, as you can see here in this diagram, the interphase is the G1, S and G2 phase. So checkpoints occur at G1 phase, I have already told you, S phase as well as G2 phase, and also at M phase. Got it? So these checkpoints are nothing but surveillance system to ensure that the interphase is completed in a proper way and the cell enters into various stages of interphase as well as into the M phase in a well maintained genetic material, so that they can be transformed, they can be transferred to the next generation. Okay, I hope this video will be beneficial to you and will be helpful to you in preparing for your forthcoming examination. Thank you.